John Craig Venter, born October 14, 1946, is an American biotechnologist, biochemist, geneticist, and entrepreneur. He is known for being one of the first to sequence the human genome and the first to transfect a cell with a synthetic genome. Venter founded Celera Genomics, the Institute for Genomic Research, TIGR, and the J. Craig Venter Institute, JCVI, and is now CEO of Human Longevity Incorporated. He was listed on Time magazine's 2007 and 2008 Time 100 list of the most influential people in the world. In 2010, the British magazine New Statesman listed Craig Venter at 14th in the list of the world's 50 most influential figures 2010. He is a member of the USA Science and Engineering Festival's Advisory Board. Early life and education. Venter was born in Salt Lake City, Utah, the son of Elizabeth and John Venter. In his youth, he did not take his education seriously, preferring to spend his time on the water in boats or surfing. According to his biography, A Life Decoded, he was set to never be a terribly engaged student, having C's and D's on his 8th grade report cards. He graduated from Mills High School in Millbrae, California. Although he was against the Vietnam War, Venter was drafted and enlisted in the United States Navy, where he worked in the intensive care ward of a field hospital. While in Vietnam, he attempted suicide by swimming out to sea, but as he got closer to the deep blue sea and was approaching the circling of a shark, he changed his mind more than a mile out. Being confronted with wounded, maimed, and dying on a daily basis instilled in him a desire to study medicine, although he later switched to biomedical research. Venter began his college education at a community college, College of San Mateo in California, and later transferred to the University of California, San Diego, where he studied under biochemist Nathan O. Kaplan. He received a B.S. in biochemistry in 1972, and a Ph.D. in physiology and pharmacology in 1975, both from UCSD. He married former Ph.D. candidate Barbara Ray. After working as an associate professor, and later as full professor, at the State University of New York at Buffalo, he joined the National Institutes of Health in 1984. In Buffalo, he divorced Dr. Ray Venter and married his student, Claire M. Frazier, remaining married to her until 2005. In late 2008 he married Heather Kowalski. They live in La Jolla outside San Diego, California, where Venter got renovated a $6 million home. Venter is an atheist. Venter himself recognized his own ADHD behavior in his adolescence, and later found ADHD-linked genes in his own DNA. Career Est Controversy. While an employee of the NIH, Venter used a technique for rapidly identifying all of the mRNA strands present in a cell, and he began to use it to identify genes which are expressed in the human brain. The short cDNA sequence fragments discovered by this method are called expressed sequence tags, or ESDs. The NIH Office of Technology Transfer and Venter decided to use these ESDs in an attempt to patent the genes they identified based on their studies of mRNA expression in the human brain. When Venter disclosed this strategy during a congressional hearing, a firestorm of controversy erupted. The NIH later stopped the effort and abandoned the patent applications it had filed, following public outcry. Human Genome Project main article, Human Genome Project. Venter was passionate about the power of genomics to radically transform healthcare. Venter believed that shotgun sequencing was the fastest and most effective way to get useful human genome data. The method was rejected by the Human Genome Project, however, since some geneticists felt it would not be accurate enough for a genome as complicated as that of humans, that it would be logistically more difficult, and that it would cost significantly more. Frustrated with what Venter viewed as the slow pace of progress in the Human Genome Project, and unable to get funds for his ideas, he sought funding from the private sector to fund Celera Genomics. The goal of the company was to sequence the entire human genome and release it into the public domain for non-commercial use in much less time and for much less cost than the public Human Genome Project. The company planned to profit from their work by creating a value-added database of genomic data to which users could subscribe for a fee. The goal consequently put pressure on the public genome program and spurred several groups to redouble their efforts to produce the full sequence. DNA from 5 demographically different individuals was used by Celera to generate the sequence of the human genome. One of the individuals was Venter himself. In 2000, Venter and Frances Collins of the National Institutes of Health and U.S. Public Genome Project jointly made the announcement of the mapping of the human genome, a full 3 years ahead of the expected end of the public genome program. The announcement was made along with U.S. President Bill Clinton, and UK Prime Minister Tony Blair. Venter and Collins thus shared an award for Biography of the Year from A&E Network. On the February 15, 2001, the Human Genome Project Consortium published the first human genome in the journal Nature, and was followed, 1 day later, by a Celera publication in Science. Despite some claims that shotgun sequencing was in some ways less accurate than the clone-by-clone method chosen by the Human Genome Project, the technique became widely accepted by the scientific community. Although Celera was originally set to sequence a composite of DNA samples, partway through the sequencing, Venter switched the samples for his own DNA. After contributing to the human genome, and its release into the public domain, Venter was fired by Celera in early 2002. According to his biography, Venter was ready to leave Celera, and was fired due to conflict with the main investor, Tony White, that had existed since day one of the project. Venter writes that his main goal was always to accelerate science and thereby discovery, and he only sought help from the corporate world when he could not find funding in the public sector. Global Ocean Sampling Expedition. The Global Ocean Sampling Expedition, GOES, is an ocean exploration genome project with the goal of assessing the genetic diversity in marine microbial communities and to understand their role in nature's fundamental processes. Begun as a Sargasso Sea pilot sampling project in August 2003, Venter announced the full expedition on March 4, 2004. The project, which used Venter's personal yacht, Sorcerer II, started in Halifax, Canada, circumnavigated the globe and returned to the U.S. in January 2006. Synthetic Genomics. Venter is currently the president of the J. Craig Venter Institute, which conducts research in synthetic biology. In June 2005, he co-founded Synthetic Genomics, a firm dedicated to using modified microorganisms to produce clean fuels and biochemicals. In July 2009, ExxonMobil announced a $600 million collaboration with Synthetic Genomics to research and develop next-generation biofuels. Venter is seeking to patent the first partially synthetic species possibly to be named Mycoplasma laboratorium. There is speculation that this line of research could lead to producing bacteria that have been engineered to perform specific reactions, for example, produce fuels, make medicines, combat global warming, and so on. In May 2010, A team of scientists led by Venter became the first to successfully create what was described as synthetic life. This was done by synthesizing a very long DNA molecule containing an entire bacterium genome, and introducing this into another cell, analogous to the accomplishment of Rachard Wimmer's group, who synthesized and ligated an RNA virus genome and booted it in cell lyse 8. The single-celled organism contains 4 watermarks written into its DNA to identify it as synthetic and to help trace its descendants. The watermarks include, code table for entire alphabet with punctuations, names of 46 contributing scientists, 3 quotations, the secret email address for the cell. Individual human genome. On September 4, 2007, a team led by Sam Levy published the first complete, 6 billion letter, genome of an individual human, Venter's own DNA sequence. Some of the sequences in Venter's genome are associated with wet earwax, increased risk of antisocial behavior, Alzheimer's and cardiovascular diseases. This publication was especially interesting since it contained a diploid instead of a haploid genome and shows promise for personalized medicine via genotyping. This genome, dubbed HuRef by Levy and others, was a landmark accomplishment. The Human Reference Genome Browser is a web application for the navigation and analysis of Venter's recently published genome. The HuRef database consists of approximately 32 million DNA reads sequenced using microfluidic Sanger sequencing, assembled into 4,528 scaffolds and 4.1 million DNA variations identified by genome analysis. These variants include single nucleotide polymorphisms, SNPs, block substitutions, short and large endals, and structural variations like insertions, deletions, inversions and copy number changes. The browser enables scientists to navigate the HuRef genome assembly and sequence variations, and to compare it with the NCBI Human Build 36 assembly in the context of the NCBI and ensemble annotations. The browser provides a comparative view between NCBI and HuRef consensus sequences, the sequence multi-alignment of the HuRef assembly, ensemble and DBSNP annotations, HuRef variants, and the underlying variant evidence and functional analysis. The interface also represents the haplotype blocks from which diploid genome sequence can be inferred and the relation of variants to gene annotations. The display of variants and gene annotations are linked to external public resources including DBSNP, Ensemble, Online Mendelian Inheritance in Man, and Gene Ontology, Go. Users can search the HuRef genome using Hugo gene names, ensemble and DBSNP identifiers, HuRef TIGR scaffold locations, or NCBI chromosome locations. Users can then easily and quickly browse any genomic region via the simple and intuitive pan and zoom controls. Furthermore, data relevant to specific loci can be exported for further analysis. Human Longevity, Incorporated. On March 4, 2014, Venter and co-founders Peter Diamandis and Robert Hariri announced the formation of Human Longevity, Incorporated, a company focused on extending the healthy, high-performance, human lifespan. At the time of the announcement, the company had already raised $70 million in venture financing, which was expected to last 18 months. Venter is the Chairman and Chief Executive Officer, CEO. The company said that it plans to sequence 40,000 genomes per year, with an initial focus on cancer genomes and the genomes of cancer patients. Human Longevity's mission is to extend healthy human lifespan by the use of high-resolution big data diagnostics from genomics, metabolomics, microbiomics, and proteomics, and the use of stem cell therapy. Author of 2 books, Vinter is the author of 2 books, the first of which was ostensibly an autobiography titled A Life Decoded. 50. Vinter's second book was titled Life at the Speed of Light, in which he announced his theory that this is the generation in which there appears to be a dovetailing of the 2 previously diverse fields of science represented by computer programming and the genetic programming of life by DNA sequencing. He was applauded for his position on this by futurist Rakers Weil. In popular culture, Vinter has been the subject of several biography books, several scientific documentary books, TV documentaries, numerous magazine articles, and many speeches. Vinter has been the subject of articles in several magazines, including Wired, The Economist, Australian science magazine Cosmos, and The Atlantic. Additionally, he was featured on the Colbert Report on both February 27, 2007, and October 30, 2007. Vinter appeared in the Evolution episode of the documentary television series Understanding. On May 16, 2004, Vinter gave the commencement speech at Boston University. In a 2007 interview with New Scientist, when asked, assuming you can make synthetic bacteria, what will you do with them? Vinter replied, over the next 20 years, synthetic genomics is going to become the standard for making anything. The chemical industry will depend on it. Hopefully, a large part of the energy industry will depend on it. We really need to find an alternative to taking carbon out of the ground, burning it, and putting it into the atmosphere. That is the single biggest contribution I could make. He was on the 2007 Time 100 Most Influential People in the World list made by Time magazine. In 2007 he also received the Golden Eurydice Award for contributions to biophilosophy. On December 4, 2007, Vinter gave the de Millbrae lecture for the BBC in London. In February 2008, he gave a speech about his current work at the TED conference. Vinter delivered the 2008 convocation speech for Faculty of Science Honours and Specialization students at the University of Alberta. A transcription of the speech is available here. Vinter was featured in Time magazine's The Top 10 Everything of 2008 article. Number 3 in 2008's Top 10 Scientific Discoveries was a piece outlining his work stitching together the 582,000 base pairs necessary to invent the genetic information for a whole new bacterium. For an episode aired on July 27, 2009, Vinter was interviewed on his boat by BBC One for the first episode of TV show Bang Goes the Theory. On May 20, 2010, Vinter announced the creation of first self-replicating semi-synthetic bacterial cell. On November 21, 2010, Steve Croft profiled Vinter and his research on 60 Minutes. In the June 2011 issue of Men's Journal, Vinter was featured as the Survival Skills Celebrity of the Month. He shared various anecdotes and advice, including stories of his time in Vietnam, as well as mentioning about with melanoma upon his back, which subsequently resulted in giving a pound of flesh to surgery. Vinter is mentioned in the season finale of the first season of the science fiction series Orphan Black, a joint production of Space and BBC America. In the episode, Vinter is referenced as patenting an organism and encoding a message in the genome of that organism, an act repeated by the character of Aldous Leakey, played by Matt Frewer. While the clones fear that this renders them as nothing more than property, in reality, in the United States and Canada, where the show primarily takes place, such a patent became unenforceable due to constitutional provisions and laws against owning human beings.